cells make red blood cells, they make blood cells, hemoglobin. That's protein.
Protein, that's right.
Connected together.
That's right. Yeah, so blood cells actually do not make hemoglobin. That's one thing which is different in case of red blood cells. Red blood cells do not synthesize anything. All other cells of the body, they synthesize something, except red blood cells, because they do not have any nucleus in them. So they are the only cells in our body that do not have nucleus in them.
So how does blood form? I might be way ahead of myself. Bone marrow is all part of
That's
that.
right. It's made in the bone marrow.
Right.
So bone marrow, when the cells are made, it has everything. When it is synthesized, it has nucleus in it. But later on, through development, it's just taken out because cell does not really need any nucleus in red blood cell. The whole purpose of the red blood cell is to transport gases. That's what it does: oxygen and carbon dioxide gas. Okay, so those are the three things that we need to really pay attention to from this chapter. Cells are very small, right? How many different types of cells are you familiar with? You are familiar with bacteria, right? And you're familiar with viruses too. Viruses have cells too, right? No, they don't.
Attached to cells.
Don't. I caught you. You are sleeping. You guys are sleeping. So viruses do not have cells in them. So the type of cell that there are three types of cells total. One type of cells are called as bacteria. Other types of cells are called archaea. So you have already done that. And the third type of cells are the cells which are eukarya. These two types of cells, and these are the three. What are these three?
Domains.
Domains, very good. These are three domains of life. These are the three types of organisms. These are the three forms of life. So archaea, bacteria, and eukarya are three forms of life. These two, bacteria and archaea, are called as prokarya. And the third one is eukarya. These two combined are prokarya or prokaryotes. And the third one is eukarya. And these two are called prokarya. Why? Because they don't have a nucleus in them. That's why they call prokarya. And eukarya means nucleus containing organisms. They contain nucleus in them. So these are the three types. And bacteria and archaea, even though they are prokaryotes, they are very different from each other. That's why they are in that big category called bacteria at the domain level. And then archaea are also very different, not only because they live in different environments, but structure is also different. Okay, so eukarya and these bacteria are only one type of cells. Archaea are only one type of cells, even though there are many different ones. There are many different bacteria, many different archaea. Eukarya are slightly different because there are four very distinct type of eukarya. Who knows the names of those four distinct type of eukaryotes?
Fungi, animals.
Animals, plants, protists. Very good. So these are protists. I will start with protists because protists are very kind of early, very primitive type of organisms. Then after that will be, I will include plants. The third ones are animals or fungi, and the fourth ones are animals. Animals and fungi are third and fourth. So these are the four very different types of eukarya, or eukaryotes they are called as. Protists are mostly single cell organisms, and all other ones are multicellular organisms. And the main difference between these two groups, prokaryotes and all of those here, is the presence of nucleus and membranes, which are internal membranes. So these ones don't have. It will become more clear later on as we talk more about it. So let's take a look at, and cells are very small. Your cells in your body are so small, you have to use microscope to see them.
I have no problem with the cubes, the size of the cubes and the square area that it takes. The smaller ones are, naturally, I think the bigger ones are more surface, right? But it's the smaller ones that.
No, because these have three-dimensional structures. If it is
That's why
a
they
flat
show it in a
surface,
cube.
yes.
Right.
So the smaller the cube is, the more surface area it has.
That is similar.
Let's talk about it briefly. So we will talk about that. It will become more clear why they actually are like that. Okay, the cells are very small. They are so small you have to use microscope to see them. And the only cell, almost the only cell that we can see with naked eye in human, there's just one type of cell that you can almost see with naked eye without the use of microscope. Anybody knows what cell is that? The egg cells. Egg cells are the largest cells of the body. So those are fairly big in size. All of the cells are very small, so they have to be seen with the help of a microscope. Here actually it puts some of those relative to each other. So atoms are very small. So this unit that you see here is called angstrom. So angstrom is very small. Angstrom is very, very small. And then after that is the smaller molecules, proteins, and then this is where the viruses are. Viruses are 100 nanometer or bigger. They are very small. Anybody knows what is a nanometer? What is a meter? What is a micrometer? So then after micro comes nanometer. So these are all smaller than meter.
Isn't there a centimeter in there? Yeah.
Okay, we can add that too. Centimeter, then micrometer, and then nano. What does micro mean? Micro means millionth. 1 millionth. So it will be like this. So this is 1 millionth of a meter. 10 to the power minus 6. So meaning, 1 micrometer is 1 millionth of a meter. So very small. If you have, this is actually 1 meter. This whole thing is 39 inches long. So this is a meter. If you actually take 1 millionth of this, that will be 1 micrometer. So it's very small. And how about nanometer? What is nano?
Big and small.
Even smaller, huh? So that one is 1 billionth of a meter. So this is 1 billionth. And then this is 1 millionth. So meaning that they are smaller. What comes after that? Anybody knows? If you go even deeper, trillionth. Let's call it gazillionth. So that one actually is trillionth, 1 trillionth of a meter.
So you can't go any smaller than that, or you can?
You can. You can go to this femto. So that's 1 trillionth of a meter, which is even smaller than an atom. So if you go higher than that, you have kilometers. So that's 1,000 meters. 1 kilometer is equal to 1,000 meters. Anyway, so this is deeper, smaller. If you go in this direction, these are smaller and smaller things. Atoms are very small. So this one angstrom, A, that you see here, it means it is 1/10 of a nanometer. So it's even smaller than this. So atoms are here, smaller than a nanometer. So this is 1 nanometer, this is here. So viruses are here, about 100 nanometer in size. And then cell, which is bacteria, is about one micrometer. This symbol that you see here, this is the symbol for micro. For nano, it's N, and for micro, it is this symbol, this Greek letter, whatever it's a million. Micro mu is called mu, so that's million. If you go towards these structures here, like a normal cell in your body is about 10 micrometer in size, 10 micrometer. Egg cell is about somewhere between 100 to 200 micrometer in size. And then frog cells are much bigger. Frog egg cells are even 1 millimeter, so that you can actually see them. Has anybody seen frog eggs? Have you ever kind of played when you were little kids outside? And your parents kept on telling you not to do that, but you still kept on doing that. So have you seen frog eggs? Yeah, they're pretty big size, right? You can even see those tadpoles developing inside the eggs, even with your naked eye, without really any help. So because they're this size, so these type of things, anyway, from here to here is the capacity of your eyes. You can see those things. When things are this big, about 150 to anything bigger than that, you can see those things with naked eye. Anything which is smaller than that, within this range here, this is the range of the microscope that we have here. Those are called light microscopes. So our microscopes can show you these things, not viruses, but we can see actually bacteria with those, even though they are very small. Anything smaller than that can be seen with these microscopes, which are very powerful, that can actually magnify things millionth of time. Those are called electron microscopes. So electron microscopes can actually show you even atoms. The first time actually when I took my class to NC State a couple of years ago, we actually saw atoms there. The first time in my life. I have been involved in science in my entire life, but they have such powerful microscopes at NC State that you can actually see atoms and molecules with them, which is amazing. You cannot just imagine. Your body is made of atoms. That's the indivisible particle. So you can see that with microscopes.
So how were you able to do that? Like what were in the slides? Like what
Some metal. They had some metal.
Oh.
So in order for you to see it, you have to be killed. Yeah.
I worked for 4 years at Stanford University building an accelerator. Just the concrete aspect of it, but I never had to see them work. It was kind of almost top secret type thing. It was big, giant, huge concrete wall, tunnel.
That's right. Those are designed for those particles to bombard and travel, right?
Right. Yeah, they spent millions of dollars.
That's right. These microscopes are very expensive. Actually, I was there last year too, and they have a new microscope now there, which is almost the size of this room. It is probably this tall, almost touching the ceiling. So that one is about $300,000 to $400,000, just one microscope. So they are expensive.
Particular microscope used for something specific?
No, I mean it's kind of a general microscope. It is in a center which is called their nanofabrication center. So when I was there last summer, what we were doing, we were making these LEDs. So there was a workshop, so I attended that workshop. So we made actually LEDs. We still have those LEDs somewhere. But they used to analyze those things because LEDs are very small. They are in somewhere in this range here, in this range here, somewhere from here to here. So they have to be seen with the help of these type of very powerful microscopes. So this is where they are in relation to each other. Think very small here, going in that direction towards ant, and others they are very visible. They are very big in size. So you don't need any aid to see those type of things. Okay, so this one is talking about the compound light microscope that we will be using in our lab. So light microscope is based on the use of light, visible light. So, but its magnification power is not very high. It can show you things about 1,000 times. So that's for light microscope. For anything, if you want to see smaller things, you'll have to go to this microscope. This is actually not this one. This one. This is one type of electron microscope. TEM is for transmission. Transmission means you can see things inside. It can show you things which are inside of the cell. Inside of the cell, there are lots of things in it, so it can show you things which are inside of the cell. That's why it's called transmission. Electrons, it is based on a bombardment of electrons. So most of the times things are killed. You know, I kind of joked about it that in order to see your own cells, those cells have to be destroyed because they are bombarded with electrons, and it can show you things inside of those microscopes. This is another microscope called SEM. This is scanning. So scanning microscopes are the one that can show you only the surface, what's on the outside of something. Transmission electron microscopes show you things inside of the cells. And scanning is only for scanning, or looking at the surface only, nothing inside. Okay, let's take a look at this short video.
What do these images have in common? They each show cells, the smallest individually functioning units of life. All organisms, from prokaryotes like bacteria and archaea to eukaryotes like animals, plants and protists, are composed of one or more cells. The two main categories of cells are prokaryotic and eukaryotic. Although these cell types have many differences, they are also similar in several ways. All cells contain genetic information in the form of DNA. They also have RNA and ribosomes, which participate in the production of proteins. The cytoplasm makes up most of each cell's volume. Surrounding each cell is a cell membrane, which is composed primarily of lipids and proteins. The cell membrane separates the contents of the cell from its surroundings. Prokaryotic and eukaryotic cells also have unique features. Most notably, eukaryotic cells have a nucleus and membrane-bounded organelles. A membrane-bounded organelle has a membrane similar to the one surrounding the cell. Prokaryotes lack organelles. On average, prokaryotic cells are also about 10 times smaller than eukaryotic cells. Prokaryotic cells are superficially similar to one another, whether they are bacteria or archaea. They're smaller than eukaryotic cells, and if you could see inside, you would not find a nucleus or other membrane-bounded organelles. In most prokaryotes, a protective cell wall surrounds the cell membrane. On the other hand, the chemistry of bacterial cells and archaean cells is quite different. For example, their membranes are made up of different types of lipids. These and other chemical differences suggest that bacteria and archaea are in different domains of life. Eukaryotic cells, such as those from animals, plants, fungi, and protists, are also similar to one another. They are all relatively large and have membrane-bounded organelles. The nucleus houses the cell's DNA. Ribosomes attached to the rough endoplasmic reticulum produce proteins, and the smooth endoplasmic reticulum synthesizes lipids. The Golgi apparatus packages materials to be secreted from the cell. Peroxisomes dispose of toxins and break down fatty acids. Mitochondria release energy from food, and the cytoskeleton aids the cell in movement and supports its structure. Despite these many similarities, a few organelles differ between animal and plant cells. In an animal cell, a lysosome is a digestive centre where debris and worn-out organelles are dismantled and recycled. Plant cells rarely have lysosomes. Digestion occurs in the central vacuole of plant cells. However, regulating turgor pressure is a more important function of the central vacuole, which changes size as the cell absorbs or loses water. Many plant cells also contain chloroplasts. These organelles use light energy to produce sugars. Finally, plant cells have a cell wall that protects the cell and determines its shape. Animal cells lack a cell wall. Even within one organism, cells can look surprisingly different. Although these four images look quite different, All show eukaryotic animal cells, so they have many of the same basic features. And all eukaryotic cells show features with their prokaryotic relatives. These similarities in cell structure are convincing evidence for the unity of life.
Okay, so on your worksheet that I gave you, take a look at page number 1 and page number 2 and see if you can recognize any of those things that you just saw in this video. Page 1 and page 2. Did she mention any of those things at all? Even though she had that British accent or Australian? Cell wall, cell membrane, nucleus, good. What else? Mitochondria, lysosomes,
The Golgi apparatus.
Golgi apparatus. Okay, so
So with the ribosomes, the protein, they're a protein, right?
ribosomes make protein.
Make protein.
They are made of protein and they make protein.
Well, yeah, but they're made of protein.
Proteins and RNA, yes.
Yeah.
So these are the things that you heard. You said cell membrane, cell wall, nucleus. So nucleus is the most obvious part of the cell. This is the most conspicuous part of the cell, most obvious one. Then you also heard about Golgi complex or Golgi apparatus. You mentioned about lysosomes. What else did you hear? Mitochondria.
Ribosomes?
Ribosomes. What else? What's that? Vacuoles, very good. Vacuole. Did you forget to mention
Central.
about what cytoplasm?
Chloroplast is it too?
Chloroplast.
What was the other cell that stored used material or and recycled it or whatever?
Vacuole, lysosomes, peroxisomes. Okay, so even these are all structures that are found in case of eukaryotes. Prokaryotes do not have these structures. Prokaryotes have only ribosomes. They have cell wall. So let's see, prokaryotes. What do they have? They have cytoplasm. They have cell membrane. They have cell wall. And then they have ribosomes. And they have nothing else. So cytoplasm, cell membrane, and then cell wall, and ribosomes. Ribosomes are also found in case inside bacterial cells or in case of archaeal cells. All other ones are found in eukaryotes. So which ones of those are just unique to plants?
Chloroplast?
Cell wall.
Cell wall. Okay, let's see, on plants only. They have other things too. Cell wall is unique to them. Not animals do not have cell wall. And they also have this chloroplast. Plants also have chloroplast. And they have all other things too, but what is more specific to them is a chloroplast and cell wall. Cell wall is found in other organisms too, but not in case of animals. So in case of animals, they have all other things, but they do not have cell wall, and they do not have chloroplast. They don't have cell wall, and they don't have any chloroplast inside. Everything else is common. So everything else, other than plants having chloroplast and cell wall, everything else is common between these organisms. Even fungi have the same things. Fungi have all of these things. They do not have chloroplast, and they have everything else. All of those things are found in them. All of these things are found in them, and all of these things are found in them also.
Well, the plants don't have lysosome though, right?
Lysosome they don't have yet. They don't have lysosomes. That's just unique to animal cells. So there's another one, centriole too. So, but at this level, don't worry about that. So this is the difference between these organisms. So the main difference is between prokaryotes and eukaryotes. So eukaryotes have all of these structures. Plants, animals, protists, and fungi have all of those structures. There are some differences. Like chloroplast is unique only for plants and protists, and we talked about, even don't worry about the lysosome part also. Everything else is common in them. But none of those things are found in case of bacteria or in case of archaea. They have only cytoplasm, cell membrane, cell wall, and ribosome. And ribosomes are common in all of these organisms. Endoplasmic reticulum, nobody mentioned that. So endoplasmic reticulum
There's a rough one and a smooth one, right?
there are two different kinds, and these are the ones which fill the entire inside of the cell. They're present all around the nucleus inside the cell. Nucleus is generally centrally located like this in the middle of the cell, deep inside. Since it's a three-dimensional structure, so this nucleus is deep inside, very well protected from outside. And all of this area is filled with those tube-like structures which are called as endoplasmic reticulum. Those are tube-like, sac-like structures inside the cytoplasm of the cell. So how about the function of these things? So cell wall protects and gives the shape to the cell. Cell membrane is for transport of things. So cell membranes control what goes on inside the cell. I'm just going to erase this part.
So how does the cell wall prevent itself from blowing up, from bursting, from
Because it's a rigid structure.
too much water?
It's a very rigid structure. It's made of cellulose. So it can expand just a little bit, but it cannot expand any further than that.
So how does it prevent it from expanding any further then?
Because it's a rigid structure, so it's kind of it supports it. It cannot go any further after that. If it, so and the cell cannot generate enough pressure from inside that it will rupture it. So the pressure is not really strong enough.
Right.
So whatever pressure is built, which is called turgor pressure, it can resist that. So cell membrane, it is for kind of regulation of what goes inside the cell or comes out of the cell. Cell wall is the one that gives the shape and strength. Nucleus, what's in the nucleus?
Genetic information.
Yes. It says DNA, which is the genetic information. Endoplasmic reticulum. So it says transport materials like proteins around the cell. So there are two functions of that. So you can say for transport and for synthesis of protein. protein is synthesized there as well on ribosomes. What is the role of Golgi? Page 2. So we can call it a shipping, packaging and shipping. Actually, it's more like a shipping and receiving dock for the cell. Things come there, and then it is transported wherever it's needed inside the cell.
So when you go back to the cell wall, in the book we talking about antibiotics or some things that prevent the cell wall from performing, from building or something? They attack,
For bacteria, so antibiotics actually are targeted toward the cell wall synthesis. If bacteria cannot make their cell walls, then they cannot divide. So antibiotics actually are used for two purposes. One is to control the synthesis of cell walls, and second is to control the synthesis of their proteins. Lysosomes, those are considered as digestive system of the cell. They break down things. Mitochondria. What's the name of mitochondria there? Powerhouses. This is where energy is generated. Ribosomes
Make proteins.
makes proteins. This is where the proteins are synthesized. Vacuoles. Page 2. Storage. Storage of things and waste products. Water is stored in vacuoles too. Chloroplast.
So does it dispose of waste too, or just it stores waste?
No, it's for storage purposes.
What disposes of waste?
What disposes them? Actually, they can expel them too from outside. But the store, different chemicals are stored there. Chloroplast.
It also recycles too, right?
What is that?
I said they also recycle with some of the waste?
Again, cells do actually recycle things. So photosynthesis. Chloroplasts are where synthesis takes place, where synthesis of carbohydrates take place. So there are two of these which are related to energy, mitochondria and chloroplast. Chloroplasts synthesize energy or carbohydrates, and mitochondria break down those glucose or carbohydrates to generate energy. So what type of energy cells need? What is the form of energy that the cells can utilize? Cells are running, right? They are like you. What is the energy that runs you? Food.
Right.
So what are you getting from the food?
Carbohydrates, protein.
Carbohydrates, proteins, nucleic acids. So, and those things are
water.
converted into energy, and water too. So those things are converted into a molecule which is called ATP. Have you ever heard about that? ATP. ATP is the chemical energy that the cells need. So all the things that you are doing, consuming things that are products of photosynthesis, you are converting that into chemical form of energy that the cells can recognize. So this information is there. These are the particles, these are the structures that you need to know, and their function. So the next couple of pages, page number 3 and page number 4, is just basically asking the same thing here. Structure, function, store material. So now you know this information here. So it's actually on page number 3, it's kind of flipping the information. So it's giving you the functions and asking you what is the name of that part of the cell that carries out those things. For example, it says store material within the cell. So the answer will be what stores things inside the cell. So that's the answer for number 1. Closely stacked, flattened sacks. Those are these, the packaging and shipping part of the cell.
So when you get to the city,
Yeah, eventually you will get to the next page.
the city, the hydraulic dam.
Is protein synthesis in the ribosome? No.
Protein synthesis is done by the ribosomes.
So the hydraulic dam would be the cell wall.
Pardon? Question number 2. That's Golgi. The structure of the Golgi, that they are in the form of pancakes, stacked pancakes like these. That's why they are, it's calling them as stacked sacks. So you can also do the cell city analogy too, or any other thing, and kind of keep on working on it. There are some questions on page number 6 and 7.
Okay, I think that's.
Okay, so all of those structures that we just talked about and the one that you have in your handout, those are the ones that you need to kind of pay attention to. The function of the cell membranes actually do many things. Cell walls protect the cells from expansion and from breaking. Cell membranes are what really controls what goes inside the cell and what comes out comes out of the cell. Those are basically gates that control the transport of things across the membrane. And cell membranes are found in both animal and plant cells, both. All cells have cell membranes. Some cells do not have cell walls, but all cells have cell membranes. So that is one characteristic which is common among all cells, no matter what type of cells they are. All of them have cell membranes, but some cells do not have cell walls. So their role is very critical. They are not only the barriers, but they also regulate what passes inside to outside or from outside to inside of the cells. Cells do communicate a lot of things. They take in things, different gases or nutrients that they need inside the cell are transported through the membrane. Their membranes are made of lipids and lipid molecules. This is a special type of a lipid molecule that has 2 fatty acid tails. Like in case of triglycerides, we saw they had 3, but this one has 2. And this one is slightly different than the other ones, called phospholipids, because it has a phosphate group part of the membrane also. So this phospholipid is what really makes their cell membrane. All cells have the same type of a structure around the cell, which is made of cell membranes. So cell membranes are pretty unique because these are in the form of double layers. There are 2 of those phospholipids which are stacked tail to tail. So this structure, which indicates that this is one molecule and this is the second molecule, and these are their tails. And they are packed in such a way that their tails are joined together. They are not tight, but they are kind of joined together. They meet here in the tail side. And the entire cell, which is mostly spherical, 3-dimensional structure, is covered with those membranes. These membranes have different characteristics in them. This part is hydrophilic, and this part is hydrophobic. So phobic means from phobia. What is phobia?
Afraid.
So that means this part is what hydro means water, and phobia means that no attraction. So there is this part of the membrane, this double membrane, which is hydrophobic, meaning it does not interact with water because it is non-polar, and water can interact only with those things which are hydrophilic, which are polar in nature. So this part is completely non-polar, does not interact with water, the two ends of that, of this phospholipid bilayer, is hydrophilic. So both ends interact with water. That's why there is a lot of water inside the cell, and on the outside of the cell, this is a lot of water also. On both sides of the cell, there is about 65 to 70% water. Inside the cell, as well as outside the cell, the concentration of water is about 65 to about 70%. And that water can interact to the outside of the cell as well as inside of the cell because of this hydrophilic part of the plasma membrane or phospholipid bilayer. It is.
Say that again. So it's not inside, but you said the middle part is again so.
This is hydrophobic. This is hydrophilic. Philic means affinity, right? Attraction. So this part can bind with water, and this part can bind with water, but water cannot really interact in this area here. It can cross, but it cannot really cannot interact with the middle part, which is highly hydrophobic. So water can cross, water, but a lot of things that cross, they actually have their own channel. They have their own proteins, so they go through those proteins. Because a lot of things things which are non-polar, for example, lipids. Lipids can cross these membranes because this part is highly hydrophobic or highly non-polar. So things can cross the middle part, which are non-polar in nature also, like oils, for example. So that's why it is designed like that. It is designed to control things. Things cannot freely go inside. Things are highly regulated when they go inside the cell. So these membranes allow only some things to cross. So these membranes allow only some things to cross, and that ability to control in this way is called as selectively permeable. That means it selects some of the things that can cross the membrane, and it rejects other things. Small things, small things, and non-polar molecules can cross the membrane. And things which are bigger in size, which are not lipid molecules or polar molecules, they cannot cross the membrane. So only few things can cross directly. What about other things? Other things can cross as well, but they are able to cross through these type of gates, and these gates are made of proteins. These gates are in the form of these channels, these holes. This is a protein, another protein. They are transmembrane proteins, meaning that they are present through these membranes. Things can cross through these tiny pores, which are designed for that purpose. For example, water has its own channel. These are called channels. So those water can cross from one side to the other, and that's how actually it maintains the level on both sides of the cell. There are many different types of proteins which are present here in the membranes of the cell, like enzymes or transport proteins, are those proteins that allow things to cross through them. And then cells are not really kind of a very free structure. They are bound with something. They are attached to something. Inside the outside of the cell, they are attached to things which are on the outside. That's how your tissues are made. Otherwise, your tissues will be just loose cells. And tissues are not loose cells. They have a structure. Whenever you see a heart, you see heart looks something like that. Its shape like that is because cells are attached to each other, and then they are attached to each other or to the outside of the cell to make the structure, so the function can be performed in those cells.
Cells bind together, the membrane separates the cell from its surroundings, so they just connect from the outside, and and that's the cell membrane prevents interaction between the two cells.
Yeah, I mean, cells are basically glued on the outside. The material on the outside of the cell is called cell matrix. And that cell matrix is in the form of cement, like glue. So they are attached to something. That's why they're not free to move. They are just held together with each other, so a structure can be made. Like in case of, for example, in mammary glands, when milk is secreted in the mammary gland, it channels that milk. Milk is a protein mostly. So when milk is secreted, it is secreted through a channel and it crosses the membrane. It is secreted in a small kind of pouch-like structure in which there are some cells. These are epithelial cells. So milk is secreted here, and then from here it crosses the membrane of the cell and then goes through to the outside of the cell, and then eventually all of this milk is collected through small ducts, and then it is expressed on the outside of the body. So it is all highly coordinated type of an activity that goes on in any function of the cells. So the function of the cell wall, it not only gives the shape, but actually keeps the shape intact also. It does not allow plant cells to burst. Nucleus. The role of the nucleus is it contains the DNA, which is the hereditary material, and that hereditary material is passed from one generation to the other. When the new cells are made, it is duplicated, then it is passed on to the next generation through sexual reproduction. Then the movement of DNA is through tiny holes which are present in the walls of nucleus. Nucleus is not completely compact structure. There are tiny gaps, there are tiny holes present in the membrane of nucleus. So things can cross those and go outside. What goes outside is actually not DNA. DNA never leaves the nucleus. It is inside the nucleus and it stays there all the time. But it can make a copy of itself in the form of RNA molecules, which are shown here. These RNA molecules can cross through these tiny pores, tiny holes, and they go outside. Outside is where ribosomes are located. There are no ribosomes here. Proteins are synthesized in the ribosomes. There are no ribosomes inside the nucleus. So that's why these RNA molecules leave nucleus, go inside the cytoplasm, which is on the outside. So everything outside the cell is in the form of that cytoplasm. What is cytoplasm? If this is the cell, this is the nucleus in the middle of the cell, this part here is all cytoplasm.
Fluid it's the fluid that's between the nucleus and the cell wall.
That's right. Nucleus and cell membrane. This whole area and all the content of all the contents that are located between the nucleus, nuclear membrane, and cell membrane. Everything which is contained inside, not inside the nucleus, not outside of the cell, but in between this area is called cytoplasm. So cytoplasm has these ribosomes. RNA is synthesized here. RNA is moved into the cytoplasm where translation or where proteins are synthesized. So ribosomes are the structures where proteins are synthesized. So this is how the proteins are synthesized. This is showing the movement of protein as it is being synthesized. RNA, which came from DNA, is being bred by this ribosome, and it is making this protein chain. Those proteins are made on the outside in the cytoplasm, inside endoplasmic reticulum. So we're just going to stop here for now, and we still have one more. We'll have to talk about this and chapter number 4 next week.